I am Dr.T.Sreelatha, Assistant Professor of Botany, Pingil Government Degree College, Vaddapelli, Varangal. So today we discuss about sporophyte evolution in bryophytes. So what is sporophyte? Um, so coming to the sexual reproduction, fertilization. So antheridium, the male sex organ is called antheridium, and the female sex organ is called archegonium, and the male sex organ produces male gametes and female sex organ produces female gametes, and male gametes fusions with one female gamete and they forms one zygote. So here the bryophytes are the zygote undergoes to so many divisions and they forms sporophyte. So sporophyte having foot, seta, capsule, and the capsule contains spores and elaters. So based on this type of characters, foot, seta, capsule, elaters, layers of the capsule, so spores, these are all characters based on the sporophyte divided into so many types. So here the sporophyte evolution. So the sporophyte evolution, here two type of theory is there. One is progressive theory and another one is regressive theory. So progressive theory means here simple to complex. So the sporophyte coming from simple to complex. So that theory, progressive theory described by Bower in 1908 and supported by Cavers and Campbell in 1940. So here simple to the sporophyte evolution based on simple to complex. So this is theory, first theory, progressive theory. So the progressive theory based on simple to complex. So first one here, the first one we can see the Rexia. So here the round shape structure, middle of the sporophyte here, middle of the round shape structure, that is called sporophyte. So here the sporophyte they did not have any foot and seta, so it is only in round shape structure and it has outer layer and middle of the part, spores are present in the middle of the sporophyte and only outer layer is present. So the outer layer protect the inner part of the spores. So this is the Rexia sporophyte. It is in round shape structure and they did not have any foot and seta. So here the Rexia, the foot and seta absent here. So only capsule is present. So the capsule attached with the thallus and absorb the water and minerals and nutrition from the thallus and to help the spores, and only outer layer is present. So and the spores are present inner side of the sporophyte. This is the first stage, Rexia stage. So coming to the second stage, so here Carcinia. So the Carcinia thallus having very small foot here. So we can see the Rexia, it is in round shape structure and they did not have any foot and seta. So coming to the Carcinia, so it has small foot. So the foot attached with the thallus and absorb the water and minerals, and it used to the capsule. And outer epithelium and inner endothelium present here. So the outer epithelium, it is only one single layer, and inner endothelium, it has so many layers, and it helps to development of spores. So this is the evaluation of the sporophyte. Coming to the Rexia. So Rexia, they did not have any foot, and coming to the Carcinia, so it has foot, small foot. So next, third stage. So here we can see the Spherocarpus. So we can see the Spherocarpus. So here we can see the clear picture here. The sporophyte divided into three parts: foot, seta, and capsule. So here the foot, small bulbous shape. So if the foot attached with the thallus and absorb the water and minerals, and it used to the seta, and seta, two or three layers are present here in vertical shape, and seta absorb the water and minerals, and it used to the capsule. So here the capsule, it is in round shape structure, and it has one outer side, one jacket layer, and spores are present in the middle of the capsule. So here calyptra also present here. So calyptra present from the foot. So we can see the development of the sporophyte in Rexia. So absent of the foot and seta. Coming to the Carcinia, so small foot is present. So third stage, coming to the Spherocarpus, foot and seta also present here, and calyptra also developed here. So then next, coming to the fourth stage of the sporophyte. So here Targenia, this is Targenia. So here here also the Targenia sporophyte divided into three parts: foot, seta, and capsule. So here the foot, very developed foot. So the foot attached with the thallus and absorb the water and minerals and nutrition, and used to the sporophyte capsule. So then second part is seta attached with the foot, and it is also absorb the water and minerals, and it used to the capsule to develop the capsule. So third part is capsule. So here the spores are here. The spores are spore mother cells are present here. Next, so one jacket layer is present in outer side of the capsule. Then fifth stage. So coming to the Markansha. So here coming to the Markansha, so the sporophyte is very more develops. So more developed here. The Markansha sporophyte is divided into three parts: foot, seta, and capsule. Foot absorb the water and minerals, and it gives to seta, and seta, it is very short and thick rows. So young seta having very thick and short cells, and mature, when the spores are mature, the seta elongated, and the seta elongated, and it pushed out of it, it pushed through the capsule, it pushed out out of the capsule through the calyptra and pseudo perianth leaves. So here we can see the picture, foot, very well developed foot, seta also present in vertical rows, and young seta having very short thin cells, and mature sporophyte having very long elongated structure, and the seta produces calyptra and pseudo perianth leaves. And capsule, the capsule, it is in round shape structure, and it has two type of spores. One is two type of spore mother cells, one is sterile mother cells, and another one is fertile mother cells. So here the sterile mother cells converted into elaters. So sterile mother cell converted into elaters. So the elaters have spiral thickenings. So the elaters have spiral thickenings. So remaining 50% fertile spore mother cells are undergoes to meiosis division, and spore mother cells are in diploid stage, so they undergoes to meiosis division, and the spore mother cells divided into 4 cells, and that stage is called tetra stage. So the tetra stage, so the tetraspores are again separate matured, coming to mature, and they separated from each other, and they forms single haploid spores. So here the Markansha development of sporophyte, here evaluation of sporophyte, that calyptra is present here, pseudo elaters are present here, and long elongated structure of the seta present here, and pseudo perianth leaves also present here. Then coming to the Markansha, next coming to the Paleo. So here coming to the Paleo, so same it is also shape of the similar to Markansha sporophyte, but here it is also divided into foot, seta, and capsule. So foot observe the attached with the thallus and observe the water and minerals, and it gives to the capsule, and second part is seta also seta attached with the foot and observe the water and minerals, and it is used to capsule. So then here that capsule, it is very developed capsule, so it has 3 to 4 layers of the jacket layers. So we can see the last pictures. So here the Markansha having only one cell of the one layer of the jacket layers. So here also Targenia also having only one layer of the jacket cells. So here Spherocarpus having only one layer of the jacket layers, and Rexia also only one layer of the jacket layer present. But coming to the Paleo, so the capsule having 3 to 4 layers of the jacket layers. So here also pseudo elaters are not pseudo elaters, only elaters are present here. Elaters are in narrow shape structure, and spore mother cells are 50% spore mother cells are converted into narrow shape structure of the elaters, and 50% spore mother cells are fertile. So the spore mother cells undergoes to meiosis division, and they forms haploid spores. So next coming to the 7th stage, so we can see the Antacid sporophyte. So here the Antacid sporophyte, it is also divided into 3 parts: foot observe attached with the thallus and absorb the water and minerals, and it gives to the capsule. And here second part is seta. Here seta absent here, so instead of seta meristematic tissue is present here. So what is the function of meristematic tissue means, so the meristematic tissue produces new cells continuously. They added new cells, they adds new cells. So the sporophytes continuously grow into more time, continuously grow into long period. So the sporophyte forms long elongated structure. So this type of sporophytes having Anthocerous thallus. So Anthocerous plants are called hornworts. So here the second part is meristematic tissue and the third part is capsule. So here the evolution of theory. So here columella is present here, 16 vertical rows, middle of the place. 16 vertical rows are present here. The parenchyma tissue made up by parenchyma tissue, that part is called columella. So here another development character here we can see here columella. So the columella present here, present in the middle of the sporophyte, middle of the capsule, and it is in present 16 rows, in 16 vertical rows made up by parenchyma tissue, and it gives to mechanical support to the sporophyte. And outer side 4 to 5 layers of epidermis present here, 4 to 5 layers. It is also development character of the sporophyte. So 4 to 5 layers of epidermis present here. So the epidermis having stomata. So the epidermis having stomata. So here the sporophyte depended on the thallus only, water and minerals. So here the sporophyte partially depended on the thallus, so only absorb the water and minerals, and only absorb the water and minerals, and they participate. So chloroplast contains chlorophyll cells, so they participates in the photosynthesis. So they prepare their own food. So here the sporophyte prepare their own food. So next here the columella present here, 4 to 5 layers of the epidermis present here, both side, and stomata also present here. Stomata contains chlorophyll cells, so they participates in the photosynthesis and prepare their own food. So here the sporophyte partially depended on the thallus only, they depended on the thallus for water and minerals. So next in between the columella and epidermis, spore mother cells are present here, and spore mother cells 50% fertile and 50% sterile. So 50% sterile mother cells are converted into here pseudo-elaters. What is meant by pseudo-elaters? It has they did not have any spiral thickenings. So they are called pseudo-elaters. So it is in narrow shape structure, and remaining 50% fertile mother cells are undergoes to meiosis division, and they divided into 4 haploid spores. That stage is called tetraspore stage, and next the tetraspores separated from each other, and they forms single haploid spores. So here what is the evolution of the sporophyte here? Progression, columella present here and epidermis present here, stomata also present here. Sporophyte only partially depended on the thallus, only depended for water and minerals, and pseudo-elatus present here. Meristematic tissue instead of seta, meristematic tissue present here. So these are all progressive characters. So next, coming to the last stage of the bryophyta. So the sporophyte, well matured sporophyte, we can see in polytrichum. So here the sporophyte very matured sporophyte here. It is also divided into foot and seta and capsule. So here apophysis, theca, operculum also present here, spore sacs also present here, spore sac having spores, and peristom teeth present here. So the spores comes outside from the peristom teeth. So columella also present here. Columella gives to mechanical support to the sporophyte. So well developed sporophyte, we can see the picture, well developed sporophyte. We can see the columella, spore sacs contains spores, and apophysis present here, theca, operculum, peristom teeth, diaphragm present here. So well developed mature sporophyte. We can see the polytrichum. Can see the polytrichum and punaria. Next, coming to the reduction theory. So we can see the progressive theory. So the sporophyte evaluated one by one. So the here reduction theory. So here reduction theory, this is the opposite of the progressive theory. So it was described by Kashyap, Church and Gamble. So coming to the regressive theory. So here complex, this theory based on the complex systems to simple one. So progressive theory simple to complex. So the regressive theory complex to simple. So here total parts. We can see the one by one. So here coming to the complex to simple one. So here first capsule here, the funaria sporophyte and polytrichum sporophyte. So then here in progressive theory, well development parts spore sacs, peristome teeth, apophysis, epiphragm, diaphragm. So columella present here. So in regressive theory, so lost their peristome teeth, epiphragm, diaphragm, spore sac, columella. So total parts are reduced. So here next coming to the coming to the reverse, coming to the reversely. So we can see the anthoceras. So lost here lost the peristome teeth. Here in we can see the anthoceras. There is no peristome teeth, there is no epiphragm, diaphragm, apophysis, only columella present here. Spore sac also absent here. So here reduced the peristome teeth, spore sac, apophysis, epiphragm and diaphragm. Next coming to the Markansha. So we can see the example, and here Paleo. We can compare the, did you understand children? We can compare the anthoceras and Paleo structure. So first we can compare the anthoceras and Paleo. So here columella absent here, 3 to 4 layers of epidermis absent here, and pseudo elatus also absent here. Meristematic tissue also absent here. So next coming to the Markansha. So we can see we compare with Paleo and Markansha. So here only one difference of the Markansha and Paleo, only the capsule outer layer. Here Markansha it has one layer, and the Paleo having 3 to 4 layers of the jocket cells. So here the jocket cells are reduced here. So here calyptra reduced and perianth leaves are reduced. Here perianth leaves reduced, so pseudo elatus reduced. We can see the Spherocarpus, perianth leaves reduced, pseudo elatus also reduced. So we can see the Rexia. So total sporophyte characters reduced here, only single capsule present here. Total foot absent here, seta absent here, columella absent, epidermis, stomata, columella, pseudo elatus, peristome teeth, epiphragm, diaphragm, total characters reduced here. So only capsule, one round shaped structure of the capsule present here. So the reduced theory. So here the regressive theory based on complex to simple one. So complex to simple one, we can see the first picture, funaria, sporophyte and polytrichum sporophyte. Then we can see the last picture, Rexia. So the sporophytes are lost their characters one by one. We can see the sporophyte lost their characters, lost their parts. So we can see the last picture, only Rexia is present. Rexia, it is in round shape structure and it has only one capsule only. So this is regressive theory and progressive theory. So progressive theory based on the simple to complex, and regressive theory based on the complex to simple. Thank you.